Okay. And don't worry, the session is being recorded and we'll be sharing the recording with you. Okay. So first, I just want to get an idea. How many of you are in college and how many of you are still studying in school? I want to know that. Anybody's in college maybe right now? First year, second year? Okay, nice. Most of you seem to be first year college. So that's nice. Just finished school, okay. Just finished college, okay. That's also pretty great. Okay, so that's great. Most of you are in college, and I hope that you'll have a brief idea of what I'm going to be teaching you. And anyways, I'll start the session now. So, can you see my screen?
Yeah, it's visible.
Okay, nice. Give me 1 minute, please. Okay, so I hope you can see my screen right now. Okay, that's great. So let me start now. So I'm Arin and I'm currently studying biological engineering in IIT Madras. I'm going to be taking today's session on our battle against COVID, which I'll be calling SARS Wars. Star Wars fans, I hope you'll notice it. Episode 1, the coronavirus menace. So how many of you had COVID or have any experience with COVID when during the pandemic? Okay, so can someone share the experience? Rima, would you like to share your experience during COVID? Okay, it was horrible and you were unable to breathe for 3 days. Okay, I think a lot of people can actually relate to that because it was quite a widespread disease. But I think what a lot of people have is the patient's perspective. And I think one more thing that could be valuable over here is a doctor's perspective. So today we're joined by Dr. Anand Nikalje. He was the head of the COVID task force at MGM Hospital, Aurangabad. So I think I'll give the mic to him now to speak about his experience during COVID. So, sir, it's all you now. The mic is yours.
Yeah, good morning all. Am I audible?
Yes, sir, you're audible.
Yeah. Thank you everyone. Thank you organizing team to give me an opportunity to talk to all young guys. See, I think you all are actually technocrats or doing some biomedical research and training in your curriculum. Uh, Actually, it is real a battle when we started thinking about the COVID way back in, I think, 2019. And initially, um, actually, it started in China, then in Singapore, and then America, and all other Western part. And we got first patient almost after 3 to 4 months of pandemic started in the world. So we are slightly aware about the disease because all the ICUs in US, Singapore, and China were completely occupied with the patients of COVID, and all ventilators and ICU beds were full. So when I started thinking about this kind of a viral infection which is spreading globally, I said ki we'll have to face this challenge managing patients of COVID someday or other. So I started whether I am prepared. If I am prepared, whether my team in my hospital is prepared or not. And I'll tell you, initially in probably in March 2020, April 2020, a lot of people actually looking at the news and not ready to work and not really actually prepared to work in the COVID pandemic. So when I took a meeting at my hospital and with local doctors, they said, Ki, why you are actually coming forward? There are a lot of government hospitals, they are actually allowed to manage these patients, and why we should put ourselves into the risk. So that was a very kind of a question which I realized that people are even medical people medicos are not ready to work. So I asked them ki if your close one gets an severe COVID infection and requires an ICU and ventilator, whether your government hospitals are really equipped and really standard to that level that they will manage this type of patient, and someday or other we'll have to prepare ourselves to fight this battle. And same we started preparation. So whenever you are actually looking at a war, unless you are prepared with the adequate manpower, adequate weapons to fight against war, and adequate understanding. So the science is important, and the risk which is involved in the war, that you have to understand. So preparation of war is very important. At that time, I think there was no much N95 mask available, no PP kits were available, but patients started coming, and we actually decided to work. When we started working with the patients initially who were suspected of COVID, a lot of healthcare workers, I can tell you the lot of junior doctors, nurses, support staff, they were actually reluctant to work because of the scared or real fear about getting infection, and a lot of people were dying in other parts of the globe. So I started taking their lectures, a kind of a motivation lecture that this is a responsibility of healthcare workers, and if you take proper precaution, we can actually get away with getting infection from the COVID. And actually, then we had a lot of meetings, discussion, and we started preparing people to work. And when a person like me, who is a quite senior in the hospital, who came forward, a lot of other people saw me that he is working, then we should work, and team building occurred initially. So actually, the challenge initially to take away the fear, anxiety, and a situation of scared situation was the first challenge which we faced. And then in first wave, there a lot of confusion about the treatment strategies. So there was no clear treatment guidelines, no effective medication was available, and then gradually every part of India got huge number of patients by the end of first wave, and the second wave was most dangerous, which we saw that many young patients got admitted with severe COVID infection. They required ventilator. Most of them could not survive. So at that point, what challenge we face is uh, first is having a adequate manpower. So adequate manpower, when I'm talking, it is in the numbers and it is a trained manpower. So parallelly when we're working, we have to train some people to take this number of patients. Then doctors, nurses, all whatever support staff, everybody was exhausting. And if somebody of the healthcare staff getting infection, then he used to take a leave for 7 to 14 days initially. So keeping manpower active, healthy, and away from the infection was a challenge. And I think at that moment of time, I realized that even the people who are sitting at home, they may not be really a trained in the healthcare worker, but they can really help us in doing a lot of counseling of the relatives, giving medications to them, timely medication to them, supporting them psychologically. So not only it's a pandemic challenge to the healthcare worker, I think society should have been involved in such a situation where manpower of healthcare was really exhausted to manage the huge number of patients. Then the problem of diagnosis, getting a RT-PCR test done at every part of country was not possible. There are very few labs initially which used to do a RT-PCR testing, and then we started a rapid testing like the RAT testing. So initially, even a lot of small tier 2 or tier 3 cities were not having facilities. So they used to send the sample to some big cities like tier 2 or tier 1 cities, and the report used to come very late. So during that period of time, there was a risk of transmitting infection to others, and because of that, people used to be more scared that what will be my report. So a lot of people, though they are asymptomatic, once report is positive, they used to be very scared of the positive report, and they used to rush to the hospital. Then I realized that the hospital cannot manage all the patients which are positive. And then we realized the importance of home isolation, and we created a lot of awareness about the home isolation, and then we asked patient to stay at home and get isolated at home. And then incidentally, because a lot of phone call used to come to us that my report is positive, what should I do? And I made a small YouTube video of 10 minutes what to do if your report is positive, and that got so viral in couple of days that a lot of people got a positive scientific information that even if my test is positive, if my saturation is more than 94, I am not having breathlessness, difficulty in breathing, I have no complaints except a fever, there is no need to rush to the hospital and get admitted. So we started giving monitoring schedule at home, and we prevented some of the admissions so that we can really serve really sick patients. Then there was a huge problem about the oxygen shortage in between, and having lack of ventilators. And actually at that time, a lot of actually innovative people, startups or some engineers came up with some kind of a artificial respirator or ventilator, which were really not workable in the critical situation of COVID patients in ICU. So I think there was a initial response, initial I think jerk response to manufacture a lot of ventilators, but this ventilator actually were not useful in a ICU patient. So we require a lot of ventilators during that time, and we got ventilators from whatever sources we could get. Oxygen shortage was a big problem, and we were really scared that if oxygen is over in the hospital, what will happen? There are 100 patients on ventilator, some patients on oxygen. What will happen to them? So some in the night hours we used to stay away, looking at the level of oxygen and the whether oxygen will receive within time or not. But fortunately, we could manage the situation, and no patient actually in our hospital uh, got problem with the oxygen shortage or ventilator shortage. But I know some instances where people died because of oxygen was over in the hospital. They were not so equipped, or they are not having enough backup storage of oxygen. And in emergency situation, when patient deteriorated and required ventilator, they said, "Okay, you can now move to other hospital," and other hospitals were full. So I know one young guy could not be saved because he died in the ambulance because of lack of oxygen. Then transport is one challenge. Transporting a COVID patient who is requiring oxygen or a ventilator to another hospital where much better facilities are available. And at that time, again, a good ambulances, a trained people who will transfer this patient was again a issue. And we should really look at better ambulances, better ambulance services in our country. And that even though COVID is over, I think we learned from the COVID that we require a lot of improvement in the healthcare infrastructure and transport facilities. Then I think there were huge lines of doing CT scans, and there was a um, familiar word that my CT score is 10, my CT score is 25 or something like that, and people used to get scared with the 10, 12, 15 numbers of the scan score, but that really doesn't matter. Actually, it was a radiological picture. But when we see a oxygen saturation is good, actually CT scan was not really giving us the severity of the illness. But huge lines in front of the scan and inability to our CT scan machines and technician to do the scans of this much of the patient. So actually, we needed a better technology which will do a scan fast, or a bedside technology where patient doesn't have to move frequently to do a CT scan or X-rays. And really, we have to have a scientific knowledge that every patient of such infection doesn't require CT scan. But at that time, people were so anxious to get a CT scan done, and people have unnecessarily did a scan and got a fear of having a severe infection. When I was managing a unit of 500 patients in one hospital where 100 patients were on ventilator, it's a huge task. So it was difficult for me to keep in touch with each and every patient. So we made a team, and the team used to give reporting on a time to time basis. And I actually at that time I used a mobile WhatsApp imaging and communication as a very effective tool. So I used to take a round when sitting at home early morning, and I used to get photos, pictures of the patient, reports of the patient, photos of the ventilator and monitors, and I could judge from the house that this patient is needs some kind of a modification in the treatment. So the technology which will actually, like we are talking to everybody on a virtual platform, this platform kind of a tele ICU or e ICU or e hospital, I think would help in such situation where one doctor cannot see 100 patient at taking a physical round, and some experienced doctor can really help a junior doctors just looking at the picture or reports sitting at home. So that kind of a technology I really use on a WhatsApp message, and I'm looking for an mobile app where I can actually two-way communication can occur in a hospital staff, doctors, and a senior staff sitting somewhere else and not in the hospital. So this were basically situation and challenges we faced. Um, Patients recovered even on ventilator, and basically most of the people had unscientific kind of attitude at that time. People started taking some kadas, some unproven medications, and and even lot of steroids, and which actually eventually led to some complications that a lot of patients had blood sugar level which is high in some patients. Few patients develop diabetes, few patients require fungal infection. So in such a critical situation of pandemic, which we hardly faced in last 50 to 100 years, I think after COVID or some pandemics during before actually freedom. So in that time, we have to keep a belief in the science, and scientific information actually help us to fight against the pandemics like these pandemics. So at that time, actually, a rational kind of a thinking is somewhere lost, what I feel. And we started taking a lot of unproven, unscientific medication, and we following a lot of techniques to uh, reduce our infection. But really, eventually, we worked only with N95 mask. Then we started with regular mask, and we stopped using PP kits, which was a big problem. That working 8 to 10 hours or more than 10 hours with a PP was a challenge. So we started using only mask, and we had good hand washing habit in all the staff, so that in spite of working in a all positive patients, all critically ill patients, we could save many healthcare workers getting active infection. We talk about the negative pressure room, positive pressure rooms. That is very ideal, but in this pandemic situation, we could not really get a good negative pressure or positive pressure rooms. So at last, I just wanted to tell you that it was a real challenge. It was a challenge. It was a physical challenge. It was a psychological challenge, and it was really a war which we faced for a couple of years. And I'm at the end, I'm really feel satisfied that in the situation where my services are required to the country or society, I could able to deliver it. We used to have one tagline that जिंदगी बड़ी होनी चाहिए लंबी नहीं. So that made a good message to the my colleagues and my staff that we should work, and uh, and actually um, we should fight as a responsibility on our side. Thank you. If
Thank
there,
you very much, Dr. Nikalje, for joining us. I
yeah,
think it's been 4 years since COVID, and we know a lot of things about it, what it was like in the hospitals, many of the stats, but I think there are still a lot of things that we actually don't know. And
yes,
I think
yes,
you
yes,
really brought that to light. And I think you really highlighted the kind of responsibility that a lot of people took, doctors and medical professionals, obviously, but also just common people who are just in the house. They also had a huge role to play.
yes.
And you really showed that. So thank you very much for joining us again. It was
Thank
our
you,
pleasure.
thank you. Yeah.
Thank you. Okay, so see, some of you have raised your hand. So Shiva, I see you've raised your hand. So you have something to ask. Uh, Anybody, Shiva, 24F10000113, come in. Anything. Okay. If you have any doubts, anything, feel free to put it in the chat and we'll answer it. And so I'll be starting the session now. And just a reminder, the meeting is being recorded. And at any point, if you feel that I'm going a little bit too fast or a little bit too slow, or you didn't hear something, feel free to tell me and I'll repeat it for you. So, I'm sorry, 1 minute. Okay, so I hope you can see my screen. So what I'll be covering in this session, our battle against COVID, it will be mostly to do with the detection immunization mechanisms behind during the pandemic. But I think if you have to understand that, we'll have to get a basic idea of genetics. So by that I mean DNA, RNA, proteins, their structure, functions. We need to know that. So I want to know how many of you are familiar with DNA, RNA? Anybody? Okay. Okay, that's nice. Um, so, Navya, could you share something about what you know? Just 1 minute. I'm sorry. Okay, so even if you don't know too much about DNA, it's fine because I'll be giving a brief idea of genetics anyways. So we'll start with the famous molecule DNA. So it's a molecule that basically tells the body how it's supposed to be. It's a nucleotide which consists of many, it's a polymer which consists of many nucleotides. Now, what is a nucleotide? It's a unit which has three components. It has a sugar, a phosphate, and a nitrogenous base. Now, this nitrogenous base, it is of four types. You have adenine, thymine, cytosine, and guanine. Now, these can be arranged in any sequence in the DNA, and this sequence it varies from person to person, and that's what gives each person their unique characteristics. So, you'll probably be familiar with the double helix structure of DNA as given by Crick and Watson. So, as you can see, it kind of looks like a ladder over here, and there are some connecting structures over here. So, what's happening is the nitrogenous bases on both strands, they're bonding to each other through hydrogen bonding. So adenine and thymine, they bond to each other, and you'll see there are two hydrogen bonds. So there's one over here and one over here. And it's similar for cytosine and guanine. There's three hydrogen bonds, as you can see here. So what happens is these bases, they bond to each other and they ensure the strands are connected to each other, as you can see over here. So it gives the DNA stability. Okay. That's the main feature of this. It's double stranded and it has stability. And one thing I want you to keep in mind is it may code for proteins. It may give the body its characteristics, but in a cell, it remains only in the nucleus. So how is it actually communicating outside? We'll move on to DNA's. Uh, we'll move on to DNA's sibling, which is RNA. RNA stands for ribonucleic acid, and you can call it DNA's messenger in a sense. So DNA has two strands, right? So one of its strands is called the coding strands, and it gives the same structure to RNA, but there's a small difference. The base thymine, you remember, it is not actually present in RNA. You'll instead find a separate base called uracil, as you can see over here. It replaces thymine. And an additional difference is that RNA has a single strand and it is not double-stranded. Okay. So as you can see, communicates to the proteins the information of the DNA, and it is actually the more important asset. So one thing that you might be wondering is why is uracil present over here instead of thymine, and why is RNA only a single stranded acid? Does anybody know that? Ah, yes, that's right. DNA gets transcripted into RNA, and RNA form proteins and amino acids. Okay. Can anybody tell me why RNA has only a single strand and why it contains uracil instead of thymine? Okay, it's fine. Wait. Okay, that is correct. Raki Shailaja, that is the correct reason. If it's double stranded, it will code for multiple proteins, and yeah, the process does get complicated. And there's one more reason I'll leave you to think about. RNA is much less stable than DNA. And the reason is that DNA, it has to store genetic information. So you would require a stable molecule for that. But RNA, it continuously receives information from DNA and then it has to code for proteins after that. You can't give it a stable structure because otherwise it can't be this flexible. So that's why it is more labile, it's more reactive, so that it can undergo mutation easily. That's the second reason. But yeah, okay, that's nice. So, okay. And there's one unfortunate thing about RNA. Mostly when you're talking about scientists, researchers, or even biology students, they'll mostly just talk about DNA and ignore RNA, which is pretty sad. It's kind of the forgotten sibling. So I think this is how RNA must be feeling, because DNA just at the end of the day gets more credit. Okay. So we can get to the third part now of genetics. That's proteins. Proteins, they're actually the chemicals that run the body. So it gives the body structure. For example, you have keratin. Keratin is what actually makes up your hair and nails. And you also have enzymes like protease. Enzymes are also proteins. And protease is responsible for breaking down proteins in your digestive tracts. So how is a protein made exactly? Okay. So it consists of 20 basic molecules called amino acids. These amino acids, they're connected through peptide bonds, okay, which you can see over here. So you have two amino acids over here. They'll undergo dehydrogenation. They will lose one molecule of water, and they'll be connected through a bond over here. So we'll call this a peptide bond. And these 20 amino acids, they'll be linked in some order to form a sequence. Now, I have one question. You have a sequence in RNA, but that just contains four amino acids. But in proteins, you'll need that just contains four nitrogenous bases. But over here, you need 20 amino acids. So how can that be? Because if you just have four nitrogenous bases, you can only form four proteins. If you have, let's say, two nitrogenous bases coding, you'll get 16 proteins. So how can you get a protein from that coding for 20 amino acids? Okay, that's correct. So codons code for different amino acids. And yeah, Bala Shweta 8 and Ragi Shailaja are both right, triplet. Because if you have three nitrogenous bases, they have a total of 64 total combinations they can make. And that is sufficient to make 20 amino acids. You have repeated structures again. Two codons can code. Codons are a code of basically three nitrogenous bases in RNA. So they can code for multiple amino acids. But the point is that every amino acid can be formed from here. So that's correct. You have something called a codon, which has a triplet code. Okay, so good to see that. Hmm. Okay. Now we'll come to something called the central dogma in biology, which talks about the flow of genetic information. The flow is basically DNA stores all the information. Proteins communicate that to the RNA communicates that to the proteins, and the proteins just go all around the body and carry it out. You can kind of think about it like a football match. So you have a coach who's the DNA. He has all the correct information. He needs to translate it to the players. That translator is the RNA, and the players are the proteins. They are the ones who actually go on the field and do all the work. They are the ones who put the information to use. So, to simplify the central dogma, this is how you can think of it. DNA tells it to RNA, and RNA tells it to protein. Okay. So, great. Hmm. So I think we've got a fair idea of what genetics are. So we'll actually move on to the crust now, the core now. What is a virus? Okay. Virus, it's kind of a microorganism. It's alive inside a cell, inside a prey cell, but outside that, it doesn't really live. So it's kind of tough to define what it really is. But okay, we can call it a microorganism for the time being. And it's pathogenic in organisms, pathogenic in humans and bacteria, and eventually it will take over the cell. Okay. So what actually is the structure? You'll have an envelope. Okay, it's similar to a cell membrane in a cell. Okay, it's an envelope and it will have a protein. Okay, enclosing the genome of the virus that will contain either the RNA or the DNA of the virus. Okay, they'll only have one of these two. And what happens is you have these envelope proteins over here. They will bind to receptors on the prey cell, which will force their entry into the cell. What happens inside it is they'll enter it, they'll enter the genome of the cell, and they'll start producing their own proteins from here. So this is called the lysogenic phase, where it enters the DNA of the actual cell it's invading and it starts producing its proteins. That's called the lysogenic phase. After that, it will be entering the lytic phase, where it will be actually producing its proteins and it will cause the lysing and death of the cell, after which it will take over the remaining cells in the population. Okay. So that's what happens. If anybody is interested about the lytic lysogenic phase, I'll share a link with you, which you can read about more. You'll find it in the chat. So feel free to go on that if you're interested. So, and going on. Hmm, okay. Now we know what a virus is, but what's special about the coronavirus? Okay. So its official name is SARS-CoV-2, Severe Acute Respiratory Syndrome Coronavirus 2. Okay. It's an RNA virus. It does not have DNA. Now, can you tell me why it is an RNA virus? Why does it not have DNA? Okay, what's an advantage it holds because of that?
It can make multiple copies quickly.
Uh, Sorry, who was that again?
Me.
Oh, okay. Oh, yeah, that's correct. Iko Ekaki and Aditya Anushka engine, I think that's correct. It has ease of mutation, as I said. It is able to mutate much faster than DNA, which gives it much more flexibility. So it can spread a lot easier. Yes, that's correct. Pranish also. Okay. That's one advantage it holds. And you'll notice in viruses, they're either very deadly or they're not that fatal necessarily, but they have higher rates of they're very infectious. So why is that? I'll leave you with that question. Okay. So moving on now about coronavirus. You can tell the answer in the chat. So the reason it's called corona, it's because it has spikes on its surface, which looks like crown. So that's Latin for corona. Now what happens is wait, these spikes uh, these spikes that you can see, they'll bind to a receptor on the human cell, which we call the ACE2 receptor. After that, you know the process. It will bind to the receptor, it will force its way inside, and it will combine with human DNA. And after that, it just takes over the working. Okay. So, one question you might think of, there is something in humans that codes for this ACE2 receptor. Can we just edit this receptor out? Is that a possibility? Because it seems pretty convenient, right? If the receptor is not there, coronavirus can't infect us. So does it sound good? I'll leave you with that for now. Okay, I'll leave you with that question for now. Hmm. So you can see the spikes are here. You have the RNA inside it. Okay. Now there's just one thing. RNAs are very small organisms. So how was it possible for us to actually determine the structure? Because light, we can't just use light. It's too small for us to see. Does anybody know that? Okay, so I've seen you put the answer. Yeah, so Navya and Sampurna, yes, you're right. It's not, it has other important functions. So if you just edit it out, you won't get coronavirus, but you'll have other complications which you can't afford having. So yeah, that's not under consideration. But yeah, and some of you have also gotten correctly. Electron microscopy, that's what you're going to do. Light, its wavelength is too big to actually observe particles as small as viruses. So you use electrons, which are smaller wavelengths. So well done, Kavin, Raki Shailaja. That's correct. You'll even Sagar, you'll use electron microscopy. So if you're not aware of how that works, here's what we'll be doing. Cryo-electron microscopy. You'll be taking the virus and you'll be putting it in a liquid. After that, you'll freeze it and you'll just solidify it. After that, you'll be bombarding electrons at them. Okay. And based on their patterns, what kind of patterns they make, you can try to realize, for example, where they're being reflected. You can realize that um, there is some mass present over there. There's some charge present over there, which tells you based on the um, incoming and outcoming momentum, you can try to get a proper idea of the model. So using that, it is actually very possible to understand the structure of a virus. Not only can you have a look at the proper uh, membrane of the virus, you can have a look at the protein of the virus, but on top of that, you can look at the RNA of the virus and you can determine its exact sequence. You can determine each and every nucleotide base. Okay, that's one major advantage of cryo-electron microscopy, which it holds. Or if anybody has anything to ask, you can feel free to ask me.
Could you please explain the receptor in more details? I don't know about it.
Okay, so it's a receptor that's present on human cells, okay? And when coronavirus is attacking us, it binds to this receptor, after which it's able to enter the cell, okay? One thing that some people might say is that you can just edit this receptor out of human cells. After that coronavirus, it has no door to enter the cell, right? But the issue with that is it has some important functions. So, for example, it's important for blood pressure, and you can't just neglect that, right? You can coronavirus, you don't want to have that, but on at the same time, you can't afford risk having blood pressure issues. So it's not actually a viable solution to edit it out. Okay.
Okay, thank you.
Okay. Does anybody else want to ask something? If no, then great. Yes. Hmm. Okay. So that's more about the coronavirus. Now, I think one word that we're all very, very, very familiar with is RT-PCR. Okay, that's the dreaded testing which was done to see if you were positive or not. You might know its full form. It stands for reverse transcriptase polymerase chain reaction. But do you actually know the mechanism for it? Okay, so we'll be having a look at that now. Okay. Before RT-PCR, we'll be starting with PCR, that's polymerase chain reaction. What happens is you'll take a strand of DNA, okay? Through alternate heating and cooling cycles, you can amplify the material in it. So how does that happen? See, you have one strand of DNA over here. By heating it, they will separate and they'll form two strands over here. Okay, you can see it. After that, you will attach a small primer, that's a small sequence of DNA to it. And after that, you'll cool it. Cooling will allow this small sequence to just extend its length and it will match the length of the initial strand. Okay. So what happened is we've gone through a cycle of heating and cooling. And from there, we just had one strand of DNA. But you see, we have two strands of DNA now, just from one cycle. We've doubled it. What we can do again is let's do it once again. Let's do one more cycle. You have this strand of DNA. It itself gives rise to two strands of DNA. And you have the second strand, it will give rise to two more pieces of DNA. So you see, we just had one strand at the beginning. We've just done 2 cycles now, and we've gotten 4 strands already. So, and you do one more cycle, you'll get to 8. After that, 16. It extends in powers of 2. So you get the idea of the amplification, the exponential rise in the number of strands. Okay, so that's the main part of PCR. It amplifies the amount of DNA, so it's actually possible to detect it. One strand you may miss it, but if you have many strands, it can be detected very easily through chemical tests. It's very visible. Okay, so just to give an idea of how what kind of effect it actually has, you have one strand of DNA, just give it 30 cycles. Okay, you will have around 1 billion strands. Okay, that's what's happening. Just 1 billion, you're extending that to 1 billion in just 30 cycles. Okay, so you've gotten an idea now. It is pretty useful, but there's one more problem now. We can't just directly use PCR testing. You wouldn't have heard it for coronavirus. So why is that? Why can't we use PCR testing for coronavirus directly? Correct. Nice to see. Um, Coronavirus is an RNA virus. Yeah, Navya and Gorija, that's correct. Hmm. And Rakesh Raja, yeah. So it's an RNA virus, so you can't actually do it. So instead, you will do something else called RT-PCR, which is reverse transcriptase polymerase chain reaction. So the conversion of DNA to RNA, that's called transcription. But the conversion from RNA to DNA, that's called reverse transcription. So you will be using an enzyme called reverse transcriptase. You'll take the you'll take the coronavirus sample, you'll put the reverse transcriptase enzyme in it. If the RNA is present in that, it will be transformed to DNA. And then after DNA, it's simple, right? I mean, I just showed you. We can amplify it easily. But there's one more question I want to ask. Yes, we can't use PCR. Okay, but why can't we have a different process from which we just take RNA and amplify that? Because, I mean, that would be much more simpler than just converting it to DNA, right? So that would be much simpler, right? Why can't we do that? Okay, so that's right. RNA is single stranded. That will lead to number one, the amplification rate is not that large, and on top of that, it will also most likely undergo fragmentation, which is absolutely undesirable. So we can't be going through that. Yeah, you're correct. It is thermally unstable. It will form secondary structures. So just remember, DNA is stable, RNA is more reactive. That's the main point you should remember. And yeah, that's the main reason why we actually cannot directly amplify RNA. We have to necessarily convert it to DNA before any kind of amplification. Okay. Yeah. So that's right. Hmm. Okay. So yeah, you've got an idea. That is what RT-PCR actually was. Okay. This is again a model of how PCR or RT-PCR will work. You just have one strand. It exponentially rises very quickly. Okay. So I think this was pretty great, but just one thing, you know, a few things about RT-PCR. First of all, you're aware that it takes a lot of time. So, and you know how widespread the pandemic was in many countries, India, USA, Brazil, Italy. So is RT-PCR really the best way to deal with it? Okay. So because I think there are certainly a few a few drawbacks that you can look at it. So that's a sneak peek for tomorrow. So is RT-PCR actually enough? Or is there something else, some other device that we can use, which is a better detector of COVID? Okay. One more thing. Today we saw the perspective of patients during COVID. We saw the perspective of doctors too during COVID. But what about the researchers who are actually developing vaccines, who are actually trying to develop some kind of medicine to treat COVID? What were they actually trying to do? So we'll be looking at the example of UC Berkeley, and finally, the major talk during lockdown when are we going to actually get a vaccine? So it actually came pretty early, but how did we actually get a vaccine? Okay, because if you look at the mechanisms, they were pretty interesting. So these are the sneak peeks for tomorrow. Okay, so this is the end of session one. Okay, this is the end of episode one, the attack of coronavirus. So tomorrow we'll be doing episode two. Uh, this was the we'll be doing the coronavirus menace. So I hope that you like this session. If anybody has any doubts, please feel free to put it in the chat box, or you can even unmute and ask me. Okay, uh, we're open to discussion. I'll be here, so you can ask me anything. Uh, um,
Will the next session be on Wednesday?
yes, it's on Wednesday, same time, 11 o'clock. And just a reminder to everyone again, it won't be on the general channel. It will be on the sessions channel. So please join it over there. The PPT will be shared. Don't worry. There will also be a few links which I will be sharing as resource materials. Please feel free to go through that for your own interest. And Fahad, RAT rapid antigen testing, I'll also be going through that tomorrow. It also has a few advantages, a few disadvantages. So you'll be seeing that tomorrow, on Wednesday, sorry. So don't worry. Okay. If anybody has any doubts, just again, feel free to ask me.
Sorry, I have another question. I was
No, of
going through
course.
the syllabus and I saw that there's this 3D printing activity.
Hmm. Okay, so that's actually not for this session. That's by Samiksha session for Apprentice of Immortality. So you'll have to ask her about that.
Because I was wondering, I don't have a 3D printer, so how do I take part in
Oh,
that?
oh, no, don't worry. You don't need too much knowledge beforehand. And it's fine if even if you don't have a 3D printer, you'll be able to do it effectively. That's not an issue. It's fine.
Thank you.
Yeah. I'll be attaching a few links just for your reference now. You can look at it once. Okay. Thanks a lot. It was great for me actually. So it was pretty nice to have a very interactive crowd. So I'm happy. So I hope you enjoyed it. Thanks a lot, Shilvia. Okay, I'm pretty happy. And one second. Okay, thank you. It's 3D. Yeah, it's Samiksha. Yes, it's 3D modeling and software. Yeah. Hmm, okay. There'll be a few more links. You'll find them anyways on the channel, but I'll be sharing them now in the chat if you want to look at it right now. Or if anybody is new to DNA, RNA, proteins, and they want a bit of an idea, you can look at the amino acid structure just to get a brief idea of how proteins are actually formed, what the structures are, and some basic properties of each single individual molecule. Okay. So thanks everybody. Just a reminder, the next session will be on Wednesday, 11 o'clock, and please join it at 11 a.m. sharp. Okay. Anushka, do you want to ask something? Okay. You just completed school. Is there anything you can do to catch up with the session? So, um, had you done biology in 11th and 12th? Okay, you did not. Hmm. Okay. I've attached a few links on proteins, so you can look at that once. If you want to get a basic idea of, let's say, viruses, DNA, RNA, number one, you can read a little bit from NCRT soft copy if you can find it. If not, I can even be doing it. I can share a few links with you, so don't worry about that. It's fine. I think you don't need to be afraid of being too behind or anything. You can definitely catch up well. I'll share the links with you. If you want, you can even look at NCRT, the specific sections, just to get a brief idea of DNA, RNA, and viruses. I can share that with you. So don't worry. Thank you everyone again. Thank you everyone again, and it was nice to have you. Okay. That's it. Navya, I'll be teaching most of the stuff. If I feel there's something that many people might not know, I'll be going over that in any case, and you can always ask me a doubt. So it should be fine. If you have a basic idea of even DNA, RNA as I've covered today, you should be fine, good for now. I'll be covering stuff. You can leave. Yes. Session is over. If you want to ask anything, of course, you can stay back, though. You're welcome to stay back. Shivashri, 2004 and Kamin Viswa 72. Just if you want to ask anything, please feel free. You can unmute or put it in the chat. Yeah, the bioinformatics part. So I won't give too much away, but I'll be keeping a small coding session to give you a brief idea of it. So don't worry, I'll be teaching you the relevant stuff beforehand. It will be a small coding session to give you a brief idea of it. It's on day 3, on Friday. Yeah. Yeah, it will be on browser end. Yes. If you're aware of the site, yes. Even if you're not familiar with coding, you don't have to be worried about the bioinformatics session, just so you know beforehand. Huh. Add to it 469. If it's fine if you're not a bio student, you should have a basic idea though of what I gave you today about genetics basically and viruses. So I'll be sharing the slide. You can refer to that. I've also given a few additional links. You can refer to those also. But if you've understood that, then you should be good with it. So yeah, you don't need to worry too much. And I'll be teaching you anything if I feel that you may not have done that before. So you don't need to worry. It's fine. I'm still here for any doubts. If you want, you can ask. If not, if you don't have any doubts, you're welcome to leave.